Research and Advanced Training in Virology and Vaccine Technologies. We are honored to welcome you all at this hybrid session through Zoom meetings today. We would like to express our appreciation and also gratitude to all participants here and at the Zoom meetings. Ladies and gentlemen, can you take your seats as the webinar will commence shortly. Excellencies, distinguished participants, ladies and gentlemen, we will begin our agenda today. Assalamualaikum warahmatullahi wabarakatuh. Greetings to all of us. Om Swastiastu, Namo Buddhaya, good regards. A very good afternoon and warm welcome to the International Hybrid Webinar and Closing Ceremony, third batch of INA-COMSTECH Fellowship Program for Research and Advanced Training in Virology and Vaccine Technologies. Let's give a round of applause, ladies and gentlemen, excellencies, distinguished speakers, and ladies and gentlemen. In this very special occasion, a hybrid session through the Zoom meetings today on July 29, 2024. First and foremost, let us praise and thank God for His mercy and grace we can gather here today. Excellencies, distinguished participants, ladies and gentlemen, allow me to greet the dignitaries here with us and also through the Zoom meetings. Excellencies, Mrs. Dita Novianti, the Director for Pharmaceutical Production and Distribution. Excellency, Professor Toto Subroto, MS, Head of Research Center for Molecular Biotechnology and Bioinformatics, Pajajaran University. We would like to also welcome Mrs. Linda Kewardani from Ministry of Foreign Affairs, and also distinguished speakers, distinguished senior official of the Ministry of Health of the Republic of Indonesia and other related ministries and institutions. Ladies and gentlemen, we would like to extend our gratitude and also our appreciation for all the present in this wonderful occasion. I am Santi Pradayini, and it is a great honor for me to be your MC in this afternoon occasion. Before entering the core of this event, respectfully to all invitees to stand up together for singing the Indonesian anthem, Indonesia Raya. Distinguished participant, please arise. attendees are welcome to sit back. Excellencies, distinguished participants, ladies and gentlemen, before starting today's event, let us pray together for the success of this event. Prayer will be based on individual belief. Prayer begin. Prayer done. Excellencies, distinguished participants, ladies and gentlemen. To continue our agenda today, we will enter the project presentations, and there is a special announcement for this project presentation that the committee has prepared for special gift for 5 participants who asked in the discussion session. So let's be focused on this project presentation. And to guide this session, I respectfully invite Mr. Muhammad Yusuf, PhD, to kindly to the stage. Please welcome Mr. Yusuf. Please give a round of applause.
Right, let me stand up. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon, everyone, excellencies, all participants, speakers, excellencies from Ministry of Health, Ministry from Foreign Affairs, and all invited guests from all institutions. So this is my honor to moderate the seminar, the webinar, talking about the epitope-based design for vaccine, diagnostic and therapeutics that will be presented by the international speakers from OIC countries from our event on OIC COMSTECH Training Fellowship 2024. So without further ado, I would like to invite the first 2 groups to come forward to present your excellent presentation, yeah, and deliver it to us. So yeah, please come forward, the first and second group. And please give a big applause to the first 2 groups. Yeah, I would like to introduce that all the work that presented here is a result of, in general, like 4 weeks training in Indonesia, which conducted in Bio Farma and Universitas Pajajaran. So within the past 4 weeks, they already can come up with something that interesting in terms of development of vaccine. Yeah, please have a seat. Not only vaccine and also other biotechnology product. And please have a seat. Yeah. You will see that the idea that will be presented here, the important part is that they want to inspire all of us that in mastering the technology, in developing the product, especially on vaccine, diagnostic, and therapeutics, among OIC countries is very important. So please, I would like to we would like to listen to this excellent presentation started from the first group. So you are the first group, right? Yeah. The first one is about the novel replicating vector vaccine display epitopes for multi-disease. So we hope that you can finish your presentation in 15 to 20 minutes, and will be continued by the second group, and we will open a discussion session for the participants offline and also online. So please, you can start your presentation. Thank you.
Okay, Assalamualaikum and good afternoon, everyone.
Yeah, the committee, please.
Okay, Assalamualaikum and good afternoon, everyone. So my name is Nurul Fiza Maisa. I come from University Putra, Malaysia. Here is my team. Assistant Professor Dr. Naila Shahid from the University of the Punjab, Pakistan. And we have also Dr. Muhammad Abdul Rahman Omar or Dr. Kalbi from Benadi University, Somalia. Okay, so for today, our group will present a mini project entitled Novel Replicating Vector Vaccine, displaying multi-epitope of ND and avian influenza virus. Okay, the Food and Agriculture Organization has recently released its annual report on food product for 2024. So as you can see that the report highlights a diverse landscape in the livestock sector with varying production trends across different regions. Okay, so basically for the poultry industry, okay, the US is the world's largest poultry meat producer with 70% of global output, followed by China and Brazil. And China is the world's largest egg producer, okay, with 38% of global production, followed by the US and India, about 7% each. And Asia also contribute about 40% of the global poultry, and also 64% of the egg producing region. Okay, so to meet the growing demand, world poultry meat production saw from 9 to 133 million tons between 1961 and 2020, and now if you see that from the report, it reached to the total of 146 million tons. And also the egg production shot up from the 15 to 93 millions of tons. Okay, next slide. Okay, so despite the growing of the poultry industry, okay, it is not free from the problems such as like the costing and also the emerging and re-emerging disease. So basically, there are two main diseases that infect the poultry, which is the Newcastle disease and also the avian influenza. Okay, so if you see that from this figure, there is a situation, for example, like AI, H5N1 in Asia Pacific. Okay, so this the, for example, from the world of health, from animal health. Okay, so for the AI, actually 22 countries affected, and then it caused around 5,681 outbreak, while for the ND, it's detected in 135 countries, and it affected more than 240 species of birds. And you can see that basically for the ND and AI, it will cause the morbidity and also the mortality up to 100%. And for the AI, actually is the potential zoonosis disease. And also from the CDC update, you can see that since April 2024, there are 13 human cases reported for the AI, and 9 cases were from the poultry farms. Okay, so next. Okay, so why Newcastle disease and also the AI are disease of concern? Okay, as I mentioned before, so these two diseases can cause that 100 mortality, and then can cause the mortality, can affect the poultry industry. And then for the ND, the disease has a rapid onset with the symptom between 2 and 12 days, while for the AI, it can start in a few hours, 2-3 days, okay, and up to 14 days to spread throughout the flock. So next. Okay, so you can see from this figure, so if the poultry industry production is affected by the disease, okay, so it can cause the production, it can lower the production, and of course the poultry market will be increased. Okay. Next. Okay, so what is the solution? So basically, to control the disease, the prevention and control of chicken disease is one of the most important factors for the profit and also sustainability, and also future growth, diversity, and success of the poultry farming and business. So a good disease control program basically emphasis on these 3 primary goals. As you can see, that the first one is the reduce exposure of the disease by proper sanitation and also the stress management. And then second, to increase the bird resistance by the vaccination. And then third, to treat the disease outbreak. And of course, between these 3, the vaccination is the most promising in terms of the prevention and control of the diseases. So for the vaccination, for the effective vaccination, so there is 5 main things that to make sure that the vaccination program is successful. First is the time of vaccination, okay, and then vaccine seed virus strain, again fish strain and type of vaccine, and also the type of chicken. So next. Okay, so of course, if you want to develop the vaccine, those vaccines need to meet the industrial application. For example, that easy to give to the chicken either through oral drinking water, and also it need to be cost and time effective, easy to handle, and easy to upscale. So next. Okay, so there are several platforms to develop the vaccine. So you can see that from the conventional until the next generation vaccine. And of course, the different type of platform that you need to choose, it depends on the characteristic that I have mentioned before that can able to meet the industrial requirement. Okay, so next. Okay, so with the current advance of the bioinformatics, we can actually shorten the time to develop the vaccine. What we call not referring to the any platform. Okay, so next. Okay, so in our project, we choose to use the replicating viral vector vaccine as our platform. So because it's highly safe due to host strain restriction, replicate well in vitro and in vivo, and also the stability of the foreign genetic insert grow to high titer in the cell, and also can induce the robust immune response. So next. Okay, so this is the example of the research that use the viral replicating vector. So next. Okay, so the specific objective is to design and predict functional epitope of the F and F gene from the ND and also the HA protein from the AI, and to predict the interaction between a protein target and also the small molecule, and third, to clone the epitope sequence in the non-essential gene of HA vector, and fourth, to deliver the replicating viral vector expressing the multiple epitope in the SPF chicken. And of course, the general or the long term of the objective is to develop a novel replicating vector vaccine displaying multiple epitope of F and HA protein against the Newcastle and avian influenza diseases. Next. Okay, so basically this is the overall methodology. So we have the gene, F gene, and also the HA gene from the ND and also from the AI. So we design or we predict the B cell and T cell epitope, and then we do the docking against the B cell and also T cell receptor. So and then from the result we screen and select the best epitope, okay, to clone into the HPT viruses by deleting or remove some of the non-essential gene. So next. Okay, so the viral vector of HPT, it will display the different epitope, the different combination of epitope, okay, and then we can deliver to the chicken either using the nasal spray or oral. So next. Okay, next and next. Okay, so it will eliminate the need of the injection to the chicken. Okay, so next.
Good afternoon and Assalamu alaikum. My name is Naila Shahid from Pakistan, and I am working as assistant professor in Center of Excellence in Molecular Biology, University of the Punjab, Pakistan. This is the federal government institute which is run by the Higher Education Commission of the Pakistan. So before start, first I would like to thank comstech-oic, biopharma industry, ITANA industry, Ministry of Health Indonesia, and Brain Laboratory, ITANA industry for the wonderful hospitality and great learning opportunity. So now coming to my project, first of all, thank you Dr. Fiza for the nice introduction of our project. Following the introduction, I will discuss the methodologies in which I will describe the approaches and techniques that I used to complete my research objective. So begin with the vaccine expressing B and T cell epitope. The first and most important thing is to understand the structure of the virus. So you can see in the picture, the first one is the Newcastle disease virus, the avian influenza virus, and the second one is the Newcastle disease virus. So in my project, I focused on the surface proteins because these are considered the most important target for the development of vaccine. So I used NDV-F protein as a vaccine candidate and the HA protein as a vaccine candidate for the avian influenza. These two proteins, the F protein helps in membrane fusion and the HA protein helps in the entry of the virus into the host cells. Moreover, these two proteins are highly immunogenic in nature, and these characteristics makes these proteins the best candidate for the development of the vaccine. Next. So after finalizing the protein that I could use for the vaccine development, the next task was to get the sequences of that protein. I used NCBI database server and got the protein sequences from NCBI database, and these sequences were used to predict the protein model. You can see, and these models were predicted based on the specific parameter to ensure the accurate prediction and the optimal outcome. You can see in the first picture, the GMQ score for the NDVF protein model was 0.66 and the sequence identity was 91%. And similar here, the GMQ score was almost 0.8. So if we got the GMQ score more than 0.5, the model can be considered as the best model. So after that, the next slide. After that, getting the protein model, I submitted the protein model on INA-COMSTECH database, and this database is used to predict the B cell epitopes. So the software predicted almost 33 epitopes for the F protein of NDV, and similarly almost 20 epitopes for the HA protein. But the most important was to find the best epitopes that can express on the surface of the F protein. So you can see in the picture, next slide. Next. These are the epitopes that I select, but I will explain why I select these epitopes. Next slide. You can see in the picture, the epitopes that are yellow in color, I selected these epitopes because they were accessible on the surface of the protein. So the criteria was to choose the epitopes that should be accessible on the surface, means exposed, not the buried one. So I finalized 3 epitopes from the NDV-F protein, and next slide. The 3 epitopes of the HA protein, and these epitopes were also accessible on the surface of the H protein. Next. After getting the epitope sequence, I joined these B cell epitopes with the help of linker and then dock with the chicken IgY fragment antibody to check the interaction of these epitopes with the chicken IgY. And after docking, surprisingly, I got the score was 0.9. And if we get the score like 0.7, means the interaction between 2 molecules is very strong. Then after predicting B cell epitopes, next slide. My next task was to predict the T cell epitope. But here, this was challenging for me when I was working with that software. I came to know that chicken allele, this server lacks the chicken allele. But with the help of my mentor, she suggested that you can paste the chicken MHC molecule and predict the epitopes. Then I used to predict the epitopes, and the next slide. I predict these epitopes, but for that I was not able to get their property, either these are strong binder or weak binder. So after getting their immunogenicity score, I selected the first 2 epitopes. Next slide. I checked the allergenicity and toxicity about these epitopes, but I could not find any allergic and toxic prediction about these epitopes. And the next, then I dock. Next slide. Dock the first epitopes with the MHC molecule. I was able to get almost 7 hydrogen bond interactions. So this was the indication that T cell epitopes has strong binding affinity with the MHC molecule. And similar with another epitopes. Next slide. I got almost 9 hydrogen bond interactions, and these epitopes also show me the strong bonding interaction with the MHC molecule. After finalizing, next slide, I designed a construct with the adjuvant B cell epitopes from HA and B cell epitopes from the NDV and the T cell epitopes from the NDV-F protein. And the next task was to express this vaccine construct in the herpes virus for expression of these vaccine constructs for development of non-replicating viral vaccines. And this part will be explained by Dr. Kalbi. Thank you so much.
First of all, assalamu alaikum wa rahmatullahi wa barakatuh. And good morning and good afternoon, everyone. And my name is Dr. Muhammad Abdurrahman, and from Somalia, from Banadir University also. And thanks my team, Dr. Fiza and Naila. Let's continue. In final discussion about herpes virus Turkey, and our last point in the herpes virus Turkey, also known Mardi virus, and has been since 1970 vaccine against Marcus disease. It's a member of family herpes viridae. Also MDV belong to the genus Mardi virus, include 3 serotype, and serotype 1 and 2 and 3. In Mardi virus or herpes virus Turkey, here serotype 3. HVT is frequently utilized as a vaccine factor for the expression of the heterologous antigen against a number of avian disease. HVT is several distinct benefit of vaccine factor. It is non-pathogenic to chicken, and HVT cannot spread horizontally, may prove low potential risk for releasing HVT factor. The large genome of HVT contain many non-essential regions, thus multiple foreign genes can be inserted simultaneously, making the development of multivalent vaccine possible. HVT is cell-associated viral, can induce lifelong immunity. Next. Also, Turkey herpes virus is non-pathogenic virus for domestic Turkey, and it's classified as the third serotype with the Merkel disease virus of group antigenetically and related to lymphotopic avian herpes virus. This group also include MDV serotype 1. MDV1 is pathogenic in chicken, causing cytolytic infection in B cell, latent infection in T cell, and induction of T cell lymphoma. And HVT1 is non-pathogenic in chicken, but it does induce a viremia with associate with the induction of protective immune response against the MDV. Chicken infected with HVT become persistent and presently infected and maintain long-lasting immunity. So that association of disease, Merkel disease, cause asymmetric paralysis, one or more limbs. Next slide. Here we saw an herpes virus as a taxonomy. We have an stranded DNA and virus, also MARDI virus. Our according host and natural host in the chicken, according interaction, cell receptor and host virus interaction. You see in the capsid, the most functional protein in the HVT. The capsid has composite 60 asymmetric unit and also 16 protein. Next. You see in this our genome of Turkey herpes virus. We have, and this herpes virus have two genoma, 159 and 160. Also 99 proteins and proteins. Also HVT is very similar to MDV1 and MDV2, but have within a unique long and short genome region where homologous genes share highly degree of colonarity, and the protein share the high level of amino acid identity. Next. And next. Finally, our herpes virus Turkey have, this is our structure, and this structure of this herpes virus, and according viral replication, the viral replication have two phases, lytic replication and latent replication. In the lytic replication have different phases, first attachment, first one, number one, attachment, number two, fusion, number three, capsid, and number four, transaction into three genoma, immediate early genes. Also, this virus have three genes, in immediate early and late. So in transcription, we have immediate early gene, also early viral mRNA vaccine, mRNA host polymerase. Also consists of, and finally, assembly of virus of nuclear viral factor. And latent replication, also circular viral episome in tandem with the host cell DNA using host cell replication. Final slide. Next. This example of studies our viral replication of HVT. This is our example. Next. And our benefit, what are the benefits for herpes virus Turkey? Our benefit is vaccine against virulent MD or Merkel disease. Number two, ability to genetically modify the virus for the expression protective heterogeneous antigen. Finally, HVT may be used as a factor expression gene from the Newcastle virus disease and infection and infectious bursal pathogen disease, also bronchitic virus, and also protective systematic immune response against this pathogen. Fine, next. Welcome to.
Okay, thank you. So as you can see that we have finished on the early preliminary study about the epitope, and also that we presented the benefit of the HPT virus. So basically our future planning that we can explore this different option for epitope display by refining the current prediction and simulate the molecular behavior in chemical or biological system. So means now we are studying about the epitope prediction and the interaction with the B cell and T cell. So the next we can study also the molecular dynamic between these three things, which is the epitopes and also the viral vector and also the host itself, which is the B cell epitope. Okay, and then we also can study further, okay, the effect to the host. So this one will prepare for the prototype to be used as a vaccine for the human, and then especially for the AI. So we actually can do or can suggest some new policy, for example, like for the worker for the animal poultry farm, for the chicken poultry farm to be vaccinated because they have the high risk to expose to the AI, which is the zoonosis virus. So next. Okay, so basically for the vaccine, it can give a very huge impact. For example, it can improve the current disease prevention and also the control program, and also enhance the productivity and the sustainability of the poultry industry. And also it can improve the economic and social welfare of the community, improve animal welfare, food safety and security, and also can contribute to the country's income and revenue generation. So you can see here the comparison between the vaccinated farm and also the unvaccinated farm. So the unvaccinated farm if the farm vaccinated, it can reduce the farm loss. So next. Okay, so you can see here the simple calculation, okay, for between the vaccinated farm and also the unvaccinated farm. So basically, if you have 10,000 chicken in the farm, okay, so if there is no mortality, so you can get 40,000 USD, okay. But if you have 10% mortality, so you will get 36,000 USD. So it means that you lost about USD 4,000. So next. Okay, so you see that when we do the calculation, if the farm have 10,000 chicken and that you need 100 dose per vaccine. So this one is per bottle that we prepare is 100 dose. Okay, so if 10,000 chicken per farm, so we need 10 bottles, and then we need 2 dose, so it costs about USD 200. So it's on USD 200 is cheaper than any farm loss because you can see that 10% mortality, it can come 4,000 USD loss. So next. Okay, so we come to the end. So you can see that our previous collaboration, we have collaboration with the Purbright Institute Chicken Vaccinology Center, and then we also have some bioinformatic collaboration with the Wellcome Singer Institute, and then we also have Dr. Naila also have some collaboration with University of Peninsula and also Dr. Kalbi. So next. Okay, so this is our previous work. So you can see that publication of the Dr. Naila for the vaccines, and also for me, I have my team that we already commercialized poultry vaccine, which is about 20 years ago, starting from 1996 until 2000, until now. So basically, we start from the conventional vaccine, live attenuated and kill vaccine, and also comes to the engineered vaccine, which is the reverse genetic, and also the CRISPR-based vaccine. And then also Dr. Kalbi involved in the vaccination program. So I think all of us has a background very related to this program. With that, I would like to thank you, and then I welcome for any collaboration for all of you here. Thank you.
Yeah, let's give a very big applause again, to the first group. Yeah, it's very common for the first time. It will be very nervous. So now I'm becoming nervous because of them. But we open this with a very nice discussion that we start from the One Health vaccine. So talking about vaccine is not only talking about the human vaccine, but also talking about the animal vaccine as well, because some of the disease that, especially zoonotic, if we don't control them, it will also increase the chance of the disease can be also harmful to human life. So again, thank you for the presentation. It's very important. We will open the discussion later. But before that, we continue with the second group. So I hope you can finish the presentation within 15 minutes. So please welcome second group.
Okay, so good afternoon, everyone. And I'm Jennifer. I'm from a Research and Development Department from Biotis Pharmaceuticals Indonesia. And I'm honored to present our collaborative mini project on computational screening of peptides targeting the SARS-CoV-2 Mpro for peptide-based antiviral drug development. And I'm joined with my esteemed colleague. This one is Azima, is from University Kuala Lumpur, Malaysia. And this one is Roland, is from University of Yaoundé, Cameroon. Okay, next. So to set the stage for our research, it is important to understand the current landscape of COVID-19 pandemics. As per June 2024, the WHO still reported over 100,000 of new cases globally. So this underscores the urgent needs for the effective therapeutic strategies to mitigate and also manage the impact of SARS-CoV-2. And while several vaccine and antiviral medication have been developed, like for instance the Paxlovid and Remdesivir, the emergence of new variants still pose a significant challenge because these new variants can potentially partially evade the vaccine-induced immunity and also have show resistance towards the currently available drugs. And therefore, developing the next generation antiviral is urgent. So despite the initial success of Remdesivir and especially Paxlovid on decreasing the severity and hospitalization, the main research problem we face here is the emergence of drug resistance variants. And current study conducted by Huang identifies several mutations in the main proteases, which is the Mpro, that was recently shown to be associated with nirmatrelvir resistance, which is the active compound of the Paxlovid. So among these resistance, the E166V are the most resistant mutants for nirmatrelvir, which undermine the efficacy of these drugs and thus highlighting the critical needs for the next generation of antiviral drugs. Next. So developing the antiviral is just as crucial as developing a vaccine. So here's several critical, essential role for the antiviral, but most importantly, it is important to emphasize that antiviral is not a substitute for vaccine. Instead, they play a role as a complement to vaccination because you see, some individual may not respond well to the vaccine due to the immune system issue or other health condition. So antiviral provide an alternative treatment option. And also antiviral can bridge the gap until widespread vaccination is achieved or in region where the vaccine distribution is challenging. And furthermore, antiviral drugs are crucial for treating severe and hospitalized cases because some individual may experience the long COVID and antiviral can provide a rapid therapeutic option. So when discussing about antiviral, there are two primary approaches, which is the small molecules and also peptide-based antiviral. While the most commercially antiviral drug now are small molecules. Small molecules has a simpler and smaller structure, leading to a fewer contact points with the target protein. While this simplicity might be advantageous during the development and also production, but due to the fewer contact points with the target protein, even the minor mutation in the target can disrupt the binding and leading to resistance. But in other hand, the peptide-based antiviral has a larger and more complex structure, allowing for a higher binding affinity and specificity, making it harder for a single mutation in the target to disrupt the binding significantly. And that's why peptide-based antiviral drug is a promising approach for developing an antiviral drug in the future. So I think everybody already familiar with SARS-CoV-2 structure. It is an enveloped single-stranded positive sense RNA, and the genome, as you can see in the picture, encodes 2 proteases, which is the main proteases, the 3CL-Pro or the M-Pro, and the papain-like proteases or the PL-Pro. So upon infection, the genome will be translated into large polyprotein and 2 viral proteases. And this polyprotein initially synthesized as single large protein precursors that need to be processed to generate a functional non-structural protein. And in this case, is by the M-Pro. M-Pro is the key enzyme that necessary for the SARS-CoV-2 replication and propagation by cleaving the polyprotein into 16 non-structural protein, and one of them is the RDRP, or RNA-dependent RNA polymerase. So that's why M-Pro is considered to be a promising target for drug development. So besides the critical role of M-Pro in supporting the replication of virus, M-Pro is also considered to be a promising drug target due to its highly conservation across coronaviruses, which make it a promising target for broad-spectrum antiviral drugs. Such drug could be beneficial for treating the future potential coronavirus outbreak as well. And the conserved nature of M-Pro will reduce the likelihood of rapid resistance development. In coronavirus, like especially SARS-CoV-2, there are so many variants with several mutations in the several part of the genome. However, the M-Pro remain highly conserved despite all this variation. Implies that while mutation might occur, but it's rarely happening in this region. And lastly, M-Pro is a protease that only unique to the coronaviruses, which reduce the potential for cross-reactivity with human proteases. So to conclude all of this, that's why we use the peptide base that target the highly conserved region in the coronaviruses. Hopefully that will increase the chance of developing effective and long-lasting antiviral treatment.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Okay, I'm Azima, a lecturer from University Kuala Lumpur, Malaysia. So for this mini project presentations, I will continue the next part, which is objective. As you can see from the slides, there are 2 types of objective here for short term and long term. So for this mini project, we will focus on the short term one. Okay, for the short term objective, you focus on to analyze the sequence and structural features of M-Pro to identify potential binding sites for peptides inhibitor, and then to identify potential binding sites on M-Pro. Next is to perform allergenicity and toxicity predictions of the peptide, and followed by the predictions of the binding affinity of the peptides inhibitor to the M-Pro. For the long-term objective, it will be to assess stability and dynamic of the peptide M-Pro complexes via molecular dynamic simulation, and then followed by the optimized peptide sequences for improved binding affinity and stability, and also to conduct biological activity assays to evaluate the antiviral efficacy of the peptide. Okay, next slide. Okay, this is the overall view about the methodology. It will start with the identifying of M-Pro sequence through the protein data bank and visualizations of the protein molecule through the BioVia software, and screening of the potential peptide through the DRAM website, and predictions of the allergenicity and toxicity through the ELICATS Pro and also ToxinPred. And the last one, to investigate the protein-peptide interactions through the CAPSDoc software. Okay, next slide. Okay, for the first one, for the identifications of sequence and structural analysis of the M-Pro and wild type, also the mutant strain, as I mentioned earlier, we use the protein data bank to get the details, and then that information will be analyzed through the visualizations of the protein through the BioVia Discovery Studio software. Okay, next. Okay, this is the outcomes from the process. As you can see, here are the sequence for both Mpro wild type and also mutant strains. And there's a difference of the amino acids at the residue 166. For the wild type, we have a glutamic acid, which then replaced by the valine for the mutant type. And as you can see from the molecule features, both of them have a similar forms of features. Okay, next. Okay, from the BioVia, we also can visualize the molecules. So here we can see the active sites which show the interaction between the peptides and also the Mpro. So the analysis of the active sites is very important, especially when we would like to do the peptide docking, which we can measure the distance between amino acids of the peptides and also the protein as well. So that information will be assessed or will be used for the peptide docking using the CAPSTOCs. Okay, next. Okay, once we get the all information, now it's time to identify or to screen potential candidates for the peptides. So in this project, we use a DRAM website to identify the candidates of the peptide, which it will provide with some information about the antiviral peptide. So for our project, we decided that the peptide should have all these criteria. The first one is a plant-based, and then it also must possess antiviral activity, and the length of the peptide must be less than 30 residue. So by using the website, basically, when we enter the antiviral at the quick search, it provides us with more than 3,800 possible peptides, but we only choose around 10 peptides that meets all the criteria that we need for this mini project. Okay, next. Okay, these are the list of the potential peptides for our study. Okay, next. Okay, once we get the list of the peptide, we should perform the predictions of the allergenicity and also the toxicity. Okay, we'll start with the allergenicity predictions through the ALLECAT Pro. It will help to predict the allergenic potentials of protein based on the similarity of their 3D protein structure as well as their amino acid sequence. So for this ALLECAT Pro, we just need to transfer all the protein sequence or the peptide sequence into the system, and then they will analyze all the information and provide us with this information. As you can see, there is no strong or weak evidence that show the allergy. And so all the peptide can be proceed with the toxicity test later on. Okay, next. Okay, for the toxicity predictions, we use a toxicity, which all the sequence of the peptide were submitted in batch. And again, we required all the sequence of the peptide and insert into the website and run the analysis. And again, as you can see from the results, all the peptides show no toxin. Means it's safe to be used later on, maybe by the consumer. So once we have all these lists, by looking at the allergenicity and also the toxicity, then we proceed the analysis of these peptides to the peptide docking by the CAPSTOCs. Okay, that will be presented by my friends here.
Okay, good afternoon. I'm Darolin. I'm from the University of Yaoundé. I'm natural product chemist. So I will continue the presentation with the molecular docking study. For this study, we start by the determination of the number of interactions between the peptide and the mutant Mpro. The objective was to predict binding affinity of the peptide inhibitor to Mpro mutant. So, using the BioVar software, we determine the number of interaction, also add in the CapDoc online software. And the result obtained are given in this table. But with this result, it was impossible to choose the best peptide. That is why we decided to That is why we decided to calculate the interaction energies to find the best peptide with the best energy. For this work, we use MPL22 software to minimize conformation by first using only the backbone of the molecule and by using all atoms of the molecule. MMPSA software version 14 was used to quantify the strong interaction energy. Next. As the result, we obtain the peptide TREP 00H02 as our best peptide because it shows the low energy, whether using backbone or using all atoms of the molecule. Next. Finally, we can say that our peptide DRAMP 00H20 is a potential inhibitor that can be used to inhibit SARS-CoV-2 protease M-Pro. This peptide could form more extensive interaction with the target due to its low energy interaction to the M-Pro. Also, using this peptide, which derived from the Leona Simosa plant, the final draw will meet the halal regulation criteria. For our future work, next, we are planning to access stability and dynamic of the peptide M-Pro complex via molecular dynamic simulation to optimize peptide sequences for improving binding affinity and stability, and to conduct biological activity assays to evaluate the antiviral activity of the peptide. Next. So, we cannot finish our work without acknowledge the Organization of Islamic Cooperation, the COMSTECH organization, UNPAD, the Ministry of Health, Ministry of Foreign Affairs, and Bio Farma Indonesia. So, terima kasih. Thank you for your kind attention.
Okay. Thank you very much. And I believe that today we are still talking about the SARS-CoV-2. Maybe some people getting why we are talking about this. But you know, since the virus is very prone to mutations, so of course there is another chance of re-emerging the disease. And at the time, there is a possibility that the previous immunity will not able to handle the new mutant. So at that time, we also need therapeutics, not only the vaccine. So again, we give a big applause to the second group. Yeah, and we have a session for answer and question. So I would like to open the first chance. Maybe the participant from the floor, if you have a question, you can raise your hand and also you can move forward. And yeah, please ask your question. Yeah, please, we have the first one. Please, sir, you can, yeah. Yeah, is it working? With the committee, please. Yeah, it's working.
Okay, thank you for the chance. Good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. My name is Dodi Irawan from BRIN. So actually, I want to ask about the first research And before that, I congratulate to you all for finishing the workshop in Indonesia. So I hope that the workshop will be good for your experience. So for my question, I'm a little bit skipping when following your presentation. So in here, you said that you tried to doing some kind of a multi-display, right? Epitope.
Multi-epitope.
So in here, my question is, in here you use the HVT vector for the HVT, the herpes virus, right? Yeah, the virus, the herpes virus for turkey, if I'm not mistaken, for the for the vector, right? So in here you try to display, or maybe only like make alternative, like I don't know virus that you put inside the F region and also the HA region. F is from the ND and from the Newcastle, right? Yeah, and the HA is from the avian influenza, and try to express it together so it can making the epitope. So is that right? In here, how you can distinguish about the expression? Distinguish its expression for its epitope. Yeah, do you understand what about my question? And is this HVT, another one is the HVT vector itself. It's, is it, it's already good enough for the making those as a vector? It's a common for vector in animal, because I'm new in animal vaccine. I usually develop for the human vaccine, such as dengue. So in here, I have planned in the future that using this HVT vector as a core for the VLP, virus-like particle, so I can display on the surface of the VLP multi-epitope. So that's what I want to ask.
Yeah, if
okay.
I try to, yeah, okay. Yeah.
For us, we will develop that construct. And,
the microphone, please.
According to the methodology, we will transform both MDV, that is herpes virus that is attenuated, and the construct. And we will form these construct in the virus. Then we can replicate in the cell line like chicken cells, where it will replicate. Our construct will also replicate. Then we can purify that virus and use to develop a vaccines.
Okay.
Yeah. Is it clear?
I understand.
Yeah. And the second idea about the vector,
the
because
vector
maybe
itself, yes.
some of us are familiar with like Marek
Right.
vector, but here we learn about the new vector.
Yes,
so maybe you want to learn to explain more about it. Is it common to use that, and what is the reason why you didn't choose the other?
The other, yes.
The viral vector you used, the herpes.
Herpes.
Yeah, herpes.
Herpes is.
Yeah, is it?
Compare with the common one. Why you use herpes?
Okay, sorry, you mentioned about the HVT virus.
Yes.
Is that common in chicken or what?
Yeah, in developing animal vaccine.
Okay, so basically,
compare with the other that already commonly knows. Commonly knows.
Compared to the common
other
viral vectors that we use.
Why we use HVT?
Adenovirus.
Okay, so basically, actually there are vaccine available using the HVT, and then they are very successful. okay, in terms of what we call display is multiple antigen inside one vector. Okay, so we have against Gumboro disease, if I'm not mistaken, from the BI, Boringham, Ingraham. Okay, and then that one is a very successful story. So I think because this vector is non-pathogenic to the chicken, okay, it's isolated from the Turkey, and then it's replicating very well inside the cell line and also in the chicken. So because of the non-pathogenic and because of the potential of the replicating, okay, so that we can use this vector, okay, to deliver to deliver the antigen, okay, to the chicken. So I think that one is the good candidate for us to choose for the vaccine. Okay.
And one more.
Yeah,
okay.
please,
I'm a little
please.
bit what is lost in your presentation. Did you display the epitope itself on the surface of the HVT or somewhere else?
No.
No. Okay.
We used BioVia software to check the position of epitopes on the surface. Not all the epitopes were on the surface. Some were behind the. So I choose only that were accessible on the surface. So when protein binds, the epitope directly trigger the immune response after binding. F and HA.
You can answer.
No, I choose the epitope that were accessible on the surface of, because I predicted epitopes from that F or HA protein. So I used that were accessible on exposed, not buried.
Okay, sorry. Actually, there are two things. Okay, first we have the F and HA. So the epitope predicted from the F and HA of the viruses of the AI and NDV. That epitope will choose that on the surface, which display the surface, right? But for the epitope will be displayed on the HVT coating protein. So these are two different things. So that's why I mentioned that this one previous study between the virus, AI and NDV. Okay, the prediction is between the AI and NDV, F and H gene with the host, the chicken B cell receptor. So between these two. But the display of this epitope, predicted epitope, will be on HVT. HVT virus, because after we insert the F and HA epitope to the gene of the HVT, it will display on the coating, on the capsid.
Outside, right?
Sorry?
Yeah,
outside.
on the capsid protein.
Yeah, outside.
It's also almost like a VLP, right? Almost like a VLP.
Like VLPs.
You can display on
Sorry?
like a VLP, virus-like particle, means you display on the surface of something, the core, right? The core itself is from the HVT, right?
It depends. Yeah, it can be also something like VLP. Yep.
Okay. Yes. Okay, I understand. Okay, thank you very much.
Yeah, welcome. Okay. Any question? One more, possibly? Because in the first session we are still nervous, so a pace is a bit slow. But I'm sure the second group will be much more motivated, energetic. I mean, but don't worry. Yeah. Okay. Nevertheless, if you still have questions to the first session, we can also give you opportunity after the second session, because this is also a discussion session for all of us. So I think again we give a very big applause to the first group and the second group. And yeah, because of the time, we shall continue with the second session. But again, I would like to highlight that any of you who has questions for the first two title can also discuss it later. So thank you very much for the first two groups. Thank you so much. Okay, I would like to invite the second batch. Please come forward. Yeah. And yeah, with us here in offline and also in online session, I believe there are about 100 participants. Yeah. So yeah, please come forward. Yeah, please welcome the third and fourth group for this seminar. Okay. Yeah. So you will be the third. You are the third. Okay. Yeah. To make time is more efficient, so I would like to invite the third group to start the presentation. Yeah. Please introduce the member.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. We are from Group 3. We will present our mini project with the title is Runoff, Chimeric mRNA Vaccine for Rotavirus and Norovirus. Before we start our present, I will introduce our team. The first is Mrs. Zaira Rahman. As our leader project, she is from the Indus Hospital and Health Network for Pakistan. And the next is Abdullahi Musa Kitota. He's from Muslim University of Morogoro, Tanzania. And me, Anissa Shazal from Bio-Pharma, Indonesia. Okay, next. Okay, why rotavirus and norovirus? Rotavirus and norovirus are the most important viral causes of acute gastroenteritis in children, as sourced from the NHA. And the case of the gastroenteritis, especially for the children, the case because of the rotavirus and norovirus. So that's why we try to finding the solution how to decrease the case of the rotavirus and norovirus. Next slide. Okay, rotavirus. We talk about rotavirus. Rotavirus is common symptoms like watery diarrhea and vomiting, and is the case of gastroenteritis because of rotavirus is so highly case. And there is no specific medicine to treat rotavirus infection. And if the person infection of the rotavirus, about 2 days after a person is exposed to the virus, they all get the symptoms. What the symptoms? Next slide. The symptoms is the baby can have diarrhea, and the frequency is often and watery. And the baby can have abdominal pain, and then fever. The fever is the temperature is from the moderate until high, and then vomiting and so tiredness. And also we need to be alert if we get the symptoms like dry diapers and then crying with no tears and dry mouth and throat, because the symptoms is the symptoms of the dehydration. And the dehydration, it can make people die. Next. Okay, we'll talk about the genome. Rotavirus is double-strain arenavirus of the family Rheophididae. The virus is composed of 3 concentric cells that enclose 11 gene segments. And the outermost shell contains 2 important proteins. The first is G proteins. We call it the protein PP7. Protein PP7 outer capsid glycoproteins. It's important to immunity and serotyping. And the second is pea protein. We call it VP4. And it's important because it's capsid. The important because it's the attachment viral and into an entry into host cell. So that's why VP7 and VP4 induce neutralizing antibodies that are believed to be involved in immune protection, and they are the key target of the RNA vaccine, especially for the rotavirus. Next. And how about the cases? The cases of the rotavirus in 2016 is so many cases about the rotavirus. But now maybe the information rotavirus is reduced because we have the vaccine of the rotavirus. And especially I get the information for our country from Pakistan, Tanzania, and Indonesia. Pakistan has the 50% of children infected by rotavirus. The second, Tanzania, the 30 until 50% of children infected with rotavirus. And in Indonesia, specifically children aged 6 until 11 months, 54.2%, and the age of 12 until 23 months, 50.6%. So from this data, we get the information that the rotavirus is still constant to we get the solution for these cases. Next. And how about the prevent for the rotavirus? So, alhamdulillah, we have the rotavirus vaccine for now. The first is Rotarix. Rotarix is contain one strain, and the technology is live attenuated. The type is G1, and to given with the orally. And the oral is applicator contain latex and rubber. And the second, we have the Rotatec. The content is 5 strains. The type is G1, G2, G3, G4, and G9. And the technology is sound, live attenuated, and they administered orally. So from this vaccine, we get to know that the case of the rotavirus has decreased. And of course, so we have to, the next idea, we have to develop our vaccine based on mRNA vaccine. Next, about the norovirus, we'll to explain by Mr. Kitota.
Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you, Sister Anissa, for introducing me. I would like to introduce myself again. My name is Abdullahi Musa Kitota, a lecturer from Muslim University of Morogoro, Tanzania. Do you know Tanzania? I hope most of them you know. Now, for the part of norovirus, the viruses that are the leading cause of sporadic and acute gastroenteritis diseases in all age groups. And the vulnerable populations for this kind of infection are young children who are under 5 years of age, and also elderly population, elders who are over 65 years of age, also immunocompromised individuals. These infections accounts for 16 up to 18% of all acute gastroenteritis cases globally. And it accounts for about 685 million illnesses, and also 210,000 deaths, and also about 15 million disability annually per each year. Also, these infections most of the studies, they have reported that they occur largely in sub-Saharan African countries and also in Southeast Asia. And also, the symptoms include vomiting, nausea, abdominal pain, and cramping, pain and cramping, diarrhea, etc. So next slide. So the genome of this virus, as you can see there in the diagram, this virus comprises open reading frame, as you see there. And before this reading frame, we have they are single-stranded and also positive sense. The RNA virus found in the family Caliciviridae and the genus norovirus. So their size is about 27 nanometer. So these are this is the size, which is the particle. And here, I want to address one thing. This particulate nature of this virus and other viruses, and also other particles, including the atoms and the smallest particles that constitute the atom, are among of the particles that has been described indirectly in the Quran, in the Surah number 10, verse number 61, and also Surah number 34, verse number 3. When you go there, you find the mentioning of these particles and their wonders. The genome also has three open reading frames, which are open reading frame 1, open reading frame 2, and open reading frame 3. The open reading frame 1 encodes the non-structural proteins. There are about 7 the non-structural proteins. The open reading frame 2, just next. Open reading frame 2 and the open reading from frame 3 encodes the major structural protein, that means VP1, and the minor structural protein, which is VP2, respectively. Based on the sequences in VP1, I mean major structural protein, the genome of these viruses is divided into 10 genogroups, which are genogroup 1 up to genogroup 10. But among these genogroups, the prevalence genogroups are genogroup 1, genogroup 2, genogroup 3, genogroup 4, genogroup 6, and genogroup 7 variants. So, but among all these prevalent genogroups, the most important one, the most prevalent one is genogroup 2-4, which is a genotype of genogroup 2. Next. So the cases of this infection, in terms of prevalence, as is shown in the slides there, the global pooled prevalence is about 19.04. You can see there the sporadic cases and the outbreak cases per year. Next. The prevalence in Africa, in Tanzania, and also in Indonesia, as is seen there, as you have seen there, is in Africa, the prevalence is large. It's above 15. In most of the studies, they have reported 15 up to around 20%. And then in Tanzania, which is the country I'm come from, is about 11.44%. In Indonesia, it's about 18.47%. Next. Also, there is a co-infection between norovirus and the rotavirus. This is another aspect which we have examined. And here we have few of the prevalences from Congo, China, Tanzania, Indonesia. Congo is 12%, and China is 5%, and in Tanzania, my country is 26.03%. In Indonesia is 7.14%. But in countries where the rotavirus vaccine has been introduced, the prevalence is low. But this is the very important aspect of our study, that is sometimes you find co-infection among these two kinds of viruses. Yes, next. So the vaccine for norovirus, until now, there is no any approved vaccine. Rather, there are some of the developments to have this full approved vaccine. And these developments, there are some limitations, which, as you have addressed there, The first one is multiple variants of various genotypes. They have been detected. And these variants, sometimes you find them within a single genotype. We find some of the variants which just occur regularly, and in most cases is almost every 2 to 3 years or every 2 to 5 years you find these variants. And also another limitation is these viruses do not grow well or effectively in cell culture. So this becomes the larger hindrance, a larger barrier to the developments of the conventional vaccines. Generally, this is the second limitation. The third one is unavailability of first a standard cell culture-based neutralization assay, and also an efficient animal model. There is some of the challenge to get a well-efficient animal model to run the developments of this vaccine, and also there is an availability of trustworthy immune correlates of norovirus protective immunity to evaluate these vaccines. So for the next part, I'm inviting Dr. Zaira to continue with the, there is another slide here for me, which are the vaccine developments, the progress. Until now, there are about 4 vaccines, but at the stage of clinical trial. The first one is at the phase 2B clinical trial, and you can see there the last one is at the phase of phase 2A clinical trial, and these two are at the phase 1 clinical trial. So until now, there is no approved vaccine for these norovirus infections. Now, I welcome Dr. Zaira to continue with the next part.
Thank you. Assalamu alaikum and very good afternoon, all of you. Thank you so much, Anisa and Kijota, for such a nice overview of the rotavirus and the norovirus. So coming towards the problem statement, next slide please. So having large prevalence of rotavirus and the norovirus among the children, which lead to the morbidity and mortality. So there are some challenges still in the control of the infection, which the one is the disease burden, because still we have a lot of cases of the rotavirus and the norovirus globally. Then comes the vaccine effectiveness, because for the rotavirus, still the vaccine is available, but in some of the low middle income countries, there is unavailability of vaccine as well. And for the norovirus, we don't have any vaccine yet. And then comes the co-infection, because of when the patient infected with both the viruses, then the condition becomes worse. So all of these things will lead to the problem statement that there is a need to develop multi-epitope chimeric vaccine for the rotavirus and the norovirus. Next slide, please. So what are the advantages of having a multi-epitope mRNA-based vaccine for both the viruses? So the one thing is the cross-protection and improved immunogenicity, because we have the vaccine that targets both the viruses. The second is the economic benefits, because in terms of the vaccine development, vaccine production, vaccine administration, vaccine distribution. And then is the low morbidity and mortality, because one vaccine can provide the immunity against both the viruses. Next slide, please. So this would lead to the objective. The main objective of our study is to develop a multi-epitope mRNA-based vaccine for the rotavirus and the norovirus. So the study will be divided into 2 phases. The phase 1 includes all the computational work, which involve in the identify immunodominant conserved epitopes of rotavirus and norovirus, and the design of mRNA vaccine construct, and finally the interaction analysis of the vaccine construct with some immune cells. In the second phase, we synthesize and validate the vaccine construct in vivo, and then test the safety and the efficacy in the animal models, and finally the optimization of the vaccine formulation and the delivery method. Next slide, please. So in order to attain these objectives, here is the methodology, which starts by having selecting the sequences of the rotavirus and the norovirus. As Kitota and the Anissa has explained that for VP1 and VP2 of the norovirus and the VP7 and the VP4 are the rotavirus proteins that are the capsid proteins. So there are, we have target the group A viruses in case of the rotavirus. In case of norovirus, there are different genotypes of the norovirus that infect the humans globally. So the most prevalent genotypes are G2.2, 3, 4, 6, and 17. So the sequences, all the available sequences of all these viruses, they were downloaded, and multiple sequence alignment was done, followed by the generation of consensus sequence. This sequence will then be used for the identification of the B cell and the T cell epitopes. These B and T cell epitopes will then be further undergone screening on the basis of their toxicity profile, their allergic profile, their cross-reactivity, and on the basis of the population coverage. So the peptides that successfully passed all the filters, they were then selected to study the interaction with the MHC molecule. On the basis of the binding affinities, the vaccine construct was designed in silico, and this vaccine construct will then subjected to further for the epitope screening for all the parameters, and also determine the physicochemical properties of the vaccine construct. This vaccine construct will then be subjected to the 3D structure prediction and the model evaluation, and then this was used to study the interactions of the vaccine construct with the immune cells. Maybe for the study we used the TLR4 and TLR3, or maybe the other immune cells as well. And then MD simulation will be performed to identify the stability of the interactions between the vaccine construct and the protein. And finally, the codon optimization and the determination of the secondary structure of the mRNA, followed by the in silico immune simulation. This is done in the phase 1. In the phase 2 involve the synthesis of the mRNA construct, followed by transcription and purification of protein, and then the mRNA capping and mixing with the lipid nanoparticles, and testing the efficacy of the vaccine in vitro and in vivo models. Next slide. So on the basis of this methodology, we have some preliminary results. First, we predict the epitopes of the norovirus. So as I have mentioned in the methodology that we have selected all the major genotypes of the norovirus. So, okay, get the consensus sequence, and these are the epitopes. So there is one interesting thing, next. Next. Okay. So there is one interesting thing here. In all the genotypes, there are 2 epitopes that are pretty much conserved among all the genotypes. So, and if we check the conservation among them, so they are pretty much conserved as well in the same genotype in different sequences. So all the epitopes are non-cross-reactive to any of the human protein, and non-allergic and non-toxic. Next slide. Next. So this figure is showing the alignment of the epitopes with the sequences. Next slide. So these are the T cell epitopes. In T cell epitopes, there are only two epitopes that having some overlapping regions between G2 and G4, and they are also pretty much conserved, and they are also non-homologous to any of the human protein, non-toxic and non-allergic. Next slide. Then comes the rotavirus epitopes. In the rotavirus, these are the epitopes that are identified with a high antigenic scores. And next slide. This is showing the population coverage. So these epitopes, they are covering most of the regions of the world on the basis of the allele frequencies. Next. So on the while finalizing all the epitopes, the mRNA vaccine construct will be designed, having the cap 5' UTR, COSAC sequence for that having the start codon, then the signal peptide to ensure the synthesis of the construct, then the GPG linker to enhance the immunogenicity, then the T cell epitopes, then we link the T cell epitopes with the B cell epitopes through the KK linker, and then 6x histidine tag and 3' UTR. And these are the properties of the designed vaccine construct, which suggests that it's a stable construct. Next slide, please. So after that, codon optimization was done and the mRNA secondary structure was predicted. The energy value of -2.614 showed that it's a relatively stable construct. Next slide. So here the secondary structure and the tertiary structure of the vaccine construct was predicted, and it is validated, and which suggests that it's a stable structure. Next slide. So here is the interactions of the vaccine construct with the TLR4, and there are almost 20 hydrogen bonds with the vaccine construct and the TLR4, which shows again a stable interaction with the immune cells. Next slide, please. So these are all results of the current study. In future, there is this vaccine construct need further optimization, and we perform the MD simulations in order to assess the stability of the vaccine construct. And then comes the synthesis in the lab and encapsulation with the lipid nanoparticles. Next slide. So this is an overview that how our vaccine construct will behave and how the immune system will be generated. So it elicits all the B cells and the CD8-4 T cells and CD4 T cells as well. Next slide, please. So for the further collaboration, I will ask.
Okay, thank you, Mrs. Zaira. And the last is about the collaboration. From the primary result, we can get it is possible to continue the production. And so it's so open to collaboration with institution maybe, and then university and the company to continue this, our mini project maybe like that. And the last, runoff. Runoff is the name of our product in the next, maybe, inshallah. I hope this product can be potential and give the benefit for our consumer, because the benefit is one vaccine, you get two benefits, inshallah. And the last, for the closing, we will listen from the Mr. Kitota from the special closing.
I failed to cram. Bunga melati harum mewangi.
Cakap.
Indah menghiasi taman sari.
Cakap.
Tari makasi atasi. Atensi yang diberi. Semoga suksesi selamu mengiring. Thank you so much. We thank all the participants. We thank all the collaborations, the institutions, and so forth for this presentation, for this fellowship. May Allah rewards you, all of them, all of you. Thank you so much.
Amin, amin, ya Rabbal Alamin. Thank you so much. Yeah, and yeah, this group is very interesting. They not only come up with the idea, but they already come up with a brand. Yeah, so yeah, just hopefully the brand is, it can come true, right? Okay, yeah, now we can continue to the next group. Please, let's give a warm welcome and give a big applause to the third group, and welcome to the fourth group.
Assalamualaikum warahmatullahi wabarakatuh. My name is Bashir Mwambi. My group members are Dr. Sharif Abdulrahman Kasudi of National Research Center, Egypt, Sandugash Sanbaeva of Al-Farabi University, Kazakhstan. And this is how we collaborated to have this topic, a chimeric mRNA-based SARS-CoV-2, RSV, and influenza virus vaccine. Next. The co-circulation of respiratory viral infections by influenza, respiratory syncytial virus, and SARS is a global crisis. And before COVID-19 broke out in 2018, you see that we had influenza death and the respiratory syncytial virus death occurring. But then after influenza broke up, we continued having the death, but somehow they were neglected. So we continued having seasonal influenza, and about 5% to 10% amongst adults, 20 to 30% of children, and with about 3 to 5 million severe cases that were reported, approximately 650 deaths that were occurring annually. Then respiratory syncytial virus also continued to affect the population, and among the infants especially, and elderly. And an estimate of about 33 million cases, hospitalized 3, and about 59 cases annually reported that die. Then an emergence of COVID-19 epidemic shifted all attention to COVID-19, and somehow the developments in supporting RSV and the influenza were put to drain, and therefore these figures will show you that yes, we still have some burden, and particularly we have not had a combination of a vaccine that really tries to support all these viruses. Next. We have tried to look at some of the developments to support the infections, 3 of them, and they are about several platforms using subunit vaccines, using mRNA vaccines, using viral vector vaccines. And if you look at what, next slide, what has been done. Yes, there are quite a number that have been done. And if you look at the statuses, most of them are in preclinical development, phase 3, phase 2, phase 1, preclinical development, meaning it is still a growing field that we need to fast track and have a remedy for it. If we can try to look at SARS-CoV-2 influenza as a whole, then we shall have support to managing the infection. Next slide. So others have gone ahead to phase 2 still, phase 3, and predominantly you can see there are preclinical trials, and only one vaccine that is targeting SARS-CoV-2 influenza and the RSV has been approved by the Food and Drug Authority of the US, and this is where we probably would need to put our effort. I would want to invite my colleague to try to expound more of this.
Assalamu alaikum. I'm Sharif from Egypt. Thanks to my team members. The problem is due to the high mortality of respiratory viruses, SARS, flu, RSV, and co-infection. And the co-infection, which is called triple-demic, we found many cases of co-circulation of the three viruses together, which is causing many mortalities, especially among children and older people. And also, because of unavailability of a combined trivalent vaccine, there is a critical need for a broad spectrum and more effective vaccine capable of simultaneously targeting these viruses. Next, please. So our many projects targeting the epitope-based messenger RNA vaccine and the vaccine, our proposed vaccine composed of the three most conservative epitopes of the surface proteins of the three viruses. And we all know that the spike proteins of these viruses are more conservative and very important and play a very important role in the viral pathogenesis. So they are an ideal candidate for vaccine production. So these are the three spike protein: RBD for SARS-CoV-2, RSV, the spike protein is pre-F protein, and for the influenza virus, we targeted the hemagglutinin proteins. Next, please. So our objectives is to design a chimeric vaccine candidate targeting the three viruses using the in silico methods. The short term, we try to perform sequence analysis. We try to identify the epitopes, the most conservative epitopes of the three viruses using the NCBI gene banks. And we went through the computational studies to try to evaluate these epitopes and to make sure that these are past all criteria for conservancy and allergenicity and all the important factors. Long term, we will continue continue with computational analysis like molecular dynamics and construction of the chimeric vaccine and the lipid nanoparticle encapsulation, and we will also hoping that this many projects will go through the in vitro and in vivo assessment. Next, please. So combination of vaccines offer a strategic advantages by merging multiple antigens into a single dose and enabling the simultaneous prevention of the virus diseases. And for the messenger RNA platform we are proposing, they have many advantages, such as reducing the need for multiple injections and also decreasing the healthcare visits for both reception and guarded, simplifying vaccine management and storage, also improving the timeline and coverage vaccination. Delivery using lipid nanoparticles improves efficiency, stability, and higher temperature at room for several formulations. Also, manufacturing process for messenger RNA vaccine can be scaled up relatively easily with low cost compared to the other technologies. Next, please. Okay.
Good afternoon, everyone. In this slide, we have described the methodology we have used in our mini project. In this study, spike protein of SARS-CoV-2, hemagglutinin from the influenza R, then fusion protein from RSV used for possible epitope designing, then used immunopharmatic screening, analyzing, then molecular docking analyzing by a lot of TOPs, a lot of immunoinformatics tools. Next slide, please. You can skip this one, please. Next slide. The future work can be include structural modeling using the homology modeling techniques and refining the model using energy minimalization and the validation tools to ensure the structural integrity. Next slide, please. Then molecular docking studies, the perform of molecular docking simulations to predict the binding affinity of the chimeric vaccine to many receptors. Next slide. This step can be continued, the simulation of vaccine candidate in a physiological environment to observe their behavior and the interaction with biological molecules using computational tools. Next slide, please. From here, we have described our preliminary results. In CBI and the protein data bank used for the obtaining whole sequences molecules of targeted proteins. Next slide. The net MHC PAN server predicts bindings of our possible epitopes to the MHC receptors, and then from this server we can select the strong binding epitopes. As you can see, SARS-CoV contain 16 strong binding epitopes. Influenza contain only 3 strong binding. Then RSV pre-fusion protein include 8 strong binding epitopes. Next slide, please. From here, we have predicted of immunogenicity selected strong binding epitopes. As you can see, there are some of them had a high positive score, which means that the greater probability of eliciting in immune response. The selected positive score epitopes are used for further analyzing. Next slide, please. The next one is cross-reactivity analysis, used for the obtaining to search for human proteome to avoid the cross-reactivity. From here, from SARS-CoV spike protein, we can select only the last 2 protein sequences, peptide sequences. Then other, as you can see, other peptide sequences can be available for the further analyzing. Next slide, please. The last one was the conservancy analysis. We can see from here the percent of protein sequencing matches as identity. SARS-CoV about over the 99%, influenza about 63%, and then RSV about 97%. And all of these mentioned epitope sequences selected for the next analyzing of allergenicity and the toxicity. Next slide, please. All of these possible peptide epitope sequences are non-allergic and then non-toxic. Next slide. After the immunoinformatics analysis, we have done molecular docking. Next slide. Molecular docking for the possible identity of the binding affinity of our selected epitope sequences with MHC1 as a receptor. The selected sequences of epitopes used for further vaccine construction. Next slide, please. It's the possible vaccine construction. Total of 3 T-cell epitopes were taken for vaccine construction. Then as an adjuvant used from regarding to literature 50S ribosomal protein L3, then two types of linkers can be used to build vaccine. At the C amino terminal vaccine sequencer, you use 6 histidine. Then it's our final chimeric mRNA-based SARS-CoV-2, RSV, and influenza virus vaccine. Next slide, please. Our current project include only the in silico predictions. Then future work can be done by the gene mRNA construction, then production of mRNA, delivering formulation for delivery with the lipid nanoparticles, and conclude by the in vitro and vivo testing, including preclinical and the clinical analysis. So, it's our project. And the next slide. I'd like to say thanks for all organizers of this agenda. Thank you so much. Thank you for your attention.
Okay, let's big applause to the fourth group. Thank you very much for presenting efficiently. So, yeah, I would like to open a Q&A session for the batch. So please raise your hand if you have question. Yeah, we have one. Yeah, please. Please introduce yourself and then ask question whether to the third or fourth group. And the participant online also can ask question, and we had answer your question in the Zoom meeting as well. Yeah, please.
Thank you. My name is Sinta. You are mentioned that in mRNA-COMSTECH you use 6 times of histidine sequence. Is it for identification? And if, as we know that histidine is not safe for the vaccine, for the human vaccine, so is it only for prototype design, or it will be included in the commercial vaccine with the histidine tag in the mRNA-COMSTECH?
Okay, thank you for the question.
And
but
the second question, could I?
But to whom you address the?
Both of them, they
okay.
use the 6 times of histidine tag.
Okay.
The second question, can I? No.
Okay. You try. You can try and then. Okay. I don't know. You can ask actually the second one.
The second one is, why don't you use the 2 mRNA-COMSTECH with the full sequence of the protein rather than the epitope design? I think if you use the full sequence of protein, you will get more neutralizing epitope, more than one, that will be probably increase the neutralization and the protection.
Okay.
Thank you.
Okay, thank you. But can you repeat the first question? What was not supposed to be in the sequence that is toxic?
There is 6 times of histidine tag in the mRNA-COMSTECH in the
6 histidine tag. His tag.
Yeah.
Okay. Thank you. So the first one, who want to answer?
So in the vaccine construct, so 6 his tag is used for only for the purification of the vaccine and not in the final vaccine construct.
I think the mRNA purification is used the poly-A resin, not the histidine tag. I read in the second presentation that histidine tag is only for identification. I know that hard to have the antibody to identify the in-house construction, that not commercial one. So I think you use the histidine tag for the identification, the expression of the mRNA vaccine. So in the commercial one, you still use the 6 histidine tag, or you will cut the sequence, only the sequence that approved in the proof of concept.
Yes. Maybe in the final vaccine construct, we don't add the 6 histidine tag, but for the prototype, currently we have this option to add the 6 histidine tag for the protein identification.
Yeah, and of course there will be a lot of more discussions about this, but don't worry because all of us are nowadays are connected by online, and even after this you can also have further discussion with them. But unfortunately, because of the time, we have to finish the presentation session. And I'd like to invite all big applause to all groups that have presented their work today. And as you know, that actually all the 4 groups, they only have like 2 weeks to prepare the mini project that comes up with all these titles. And all the work has been inspiring us, again, that mastering the technology of the epitope-based vaccine design is really helpful for the country to cope with our own health problem. Yeah. So thank you again for all the participants. And with this, I would like to return the mic to you. Thank you so much.
Once again, please give a round of applause. Thank you very much to all respectful and insightful presentation and also discussion by all the speakers and also the moderator. You are pleased to return to your seat. And ladies and gentlemen, distinguished participants, we would like to continue to our next agenda with the closing ceremony. And before we will start the closing ceremony, I would like to give a little bit of announcement that in the end of the session of our today's agenda, we'll have a special game such as a Kahoot game that will be held in the end of this session. So please ensure be ready because after all this closing ceremony, we will begin the Kahoot games because all the committee already prepared 5 prize for 5 winners that already be the winner of the Kahoot game that all the committee already prepared the questions by the Kahoot game. So please prepare. Excellencies, distinguished participants, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. Once again, we would like to extend a very warm welcome to the closing ceremony of the 3rd batch of INA-COMSTECH Fellowship Program for Research and Advanced Training in Virology and Vaccine Technologies. A very special occasion and hybrid session through the Zoom meetings today, 29th of July 2024. And first and foremost, let us praise and thank God for His mercy and grace we can gather here today. Excellencies, distinguished participants, ladies and gentlemen, allow me to greet our dignitaries here with us today and also through the Zoom meetings. Excellency, Mrs. Rizka Andalusia, Director General of Pharmaceutical and Medical Devices. Excellency, Mr. Aftad Saidi, Senior Director HR and Administration of Committee for Scientific and Technological Cooperation, or COMSTECH. Excellency, Professor Rizky Abdullah, PhD, Director of Research and Community Service, Padjadjaran University. Excellency, Mr. Mr. Soleh Ayubi, PhD, Deputy Chief Executive Officer, PT Bio Farma, distinguished senior official of the Ministry of Health of Republic of Indonesia and other related ministries and institutions, ladies and gentlemen, we would like to extend our gratitude and also appreciation for the present in this wonderful occasion. And I am Santi Fredayini. It is a great honor for me to be the MC of today's event. And honorable speakers, distinguished participants, the third batch of INA-COMSTECH Fellowship Program has been implemented since July 1st, 2024, attended by 12 participants from 9 OIC member countries. All fellowship program participants had the opportunity to visit the local vaccine and biological industries, national research, and also innovation agency, also attended intensive training at PT Bio Farma and Pajajaran University. And to begin our event today, Excellencies, distinguished speakers, ladies and gentlemen, now let us hear the remarks for this important occasion from Excellency Mr. Soleh Ayubi, Deputy Chief Executive Officer of PT Bio Farma. Distinguished participants, please welcome Mr. Soleh Ayubi.
Good. Hello, Assalamualaikum warahmatullahi wabarakatuh. Good evening, Bu Rizka, and then Mr. Abdullah, and I forgot one by one, but I think distinguished panelists, and then also couple of friends from Pajajaran University, from Etana, then also, of course, from Bio Farma and all the participants. Thank you for this invitation. It's an honor for me. Bio Farma, or me at least, believes that collaboration is key for everything, right? If you take a look in the past 4 years that we have to deal with pandemic COVID-19, we learn from that event that kind of problem is too big to be tackled by only one organization, one university, one company, even one country, right? The same thing, you know, if you have to move on from the COVID-19, we still have like many healthcare challenges. We still have to deal with infectious disease, many infectious disease. We still have to deal with, you know, non-communicable disease, diabetes, high blood pressures, everything, right? Every healthcare challenges that we are currently facing, or we're gonna see like very soon down the road, that's too big to be tackled by one organization, one university, one company, or even one countries. So there is no way for us to tackle those problems by ourselves. So back to my first statement that we need collaboration, right? Bio Farma, as one of the company that has been doing a lot of work in this arena, collaboration becomes our ways of working. In the past, Bio Farma used to be Pasteur Institute. We are part of global network of healthcare organization or research organization. The same thing, if you notice, polio. I think some of you learned so much about polio, right, from Bio Farma. Polio, we started the work in 1986. There's collaboration between Bio Farma and Japanese Polio Research Institute. It's 1986, right, long time ago. And finally, we come up with that vaccine. That's become the backbone of Bio Farma, right? We're currently serving 76% of global demand on polio vaccines. That's the result of collaborations. Recently, Bio Farma has been collaborating closely with Sinovac on the COVID-19, with Baylor College of Medicine, with UNPAD, with UNER on COVID-19 Indovax vaccine. right? And we are currently doing other collaboration as well. On the HPV vaccine, we're working closely with MSD, right? And just about a month ago, we just signing collaboration with Novo Nordisk on insulin, right? So this collaboration is natural. It's the way of working of biopharma. And we believe as well, beyond that kind of industrial collaboration, working closely with universities, with UNPAD, with member from OIC, from Pakistan, from many countries, we believe that's the only way we're gonna solve healthcare challenges, which is, as you believe, it's not easy. So we're very happy to host this event. We are very happy to have you, participant from OICs, to learn so much about a couple things. I do believe that, from the background that I read from the participant, most of you are from research institute, from universities, right? And based on the CV that I got, you have like really strong on the academic, you're really strong in lab, you're really strong, I think some of you did a postdoc in the US, right? So from scientific perspective, I think you're probably even better compared to biopharma people, right? But we hope, you learn from biopharma on 3 aspects, at least. The first one is on the scaling up, from the lab setting to the manufacturing setting. I hope you learn from on that angle. The second one, I hope you learn as well the aspect of regulatory, right? You learn, so what is the important aspect of local FDA when evaluating a product. And then also I hope you learn as well on the aspect of GMP, right? Good practice manufacturing. I do believe you're even better on the GLP level, but on the GMP level, I think I hope you learn from biopharma. So today, I think the last day, and I hope you bring those knowledge to your countries. But I don't want, with the end of this event, it's also end the collaboration between your institution with biopharma. We don't want that. We want this collaboration. We want this meeting keep continue, and we're gonna working together on multiple aspects of the healthcare challenges that we're currently facing. So thank you so much. And again, we are honored. We are very happy to host you. And somehow down the road, we hope our path will cross again. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Mr. Soleil, for your remarks. And Excellencies, distinguished speakers and participants, now let us hear the remarks for this important occasion from Excellency Professor Rizky Abdullah, PhD, Director of Research and Community Service, Pajajaran University. Please welcome Professor Rizky.
Distinguished guests, esteemed colleagues, and all the participants, Assalamualaikum warahmatullahi wabarakatuh. So first of all, I would like to extend the apologies from our rector, Professor in Industry, who was planning to come to this event by herself, but today she have to attend another meeting, so she assigned me to come to this event. And I would also like to congratulate all the participants because you have shown your effort, your dedication to come to Indonesia for several weeks, leaving your family in your country. So this commitment inspires us all here in Indonesia. And this training also have provide a milestone for all of us that we now understand that the halal vaccine development has become critical now, especially among us in the Muslim countries. And for those, I would also to thanks to all the collaborators. This is not only from Pajajaran, but also with Ministry of Health, Ministry of Foreign Affairs, Biopharma, and also the IOC this collaboration between us, we can gather all the experts, and also we can create the platform in our vaccine development. And UNPAD also we are also honored to become part of this important event. And this is the testament of the dedication of our Universitas Pajajaran in the global network. And the last thing is for all the participants, now you are become part of global of the global network of professional, global network of professional in health vaccine development. So after this event, so please carry forward, please continue to carry forward the knowledge that you gain here. And when you facing some trouble or you need collaboration, please remember that we are here, your brother, sister, Indonesia. We'll be very happy to collaborate with you. And from Universitas Pajajaran, I think you already know very well Professor Toto Subroto here, Dr. Muhammad Yusuf. I'm sure they will be very kind to help you for all the problems you may face in the future. So thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you very much, Prof. Rizky, for your remarks. And Excellencies, distinguished participants, ladies and gentlemen, we would like to continue our agenda and would like to invite Mr. Muambi Bashir, MSc, PhD, as a participant representative to be able to give feedback and words for all of us about the training. Please welcome Mr. Bashir.
A'udhu billahi minasyaitonir rajim. Alhamdulillahi rabbil alamin. A'udhu billahi minasyaitonir rajim. Wassalatu wassalam ala Rasulillahi. Wassalamu alaikum warahmatullahi wabarakatuh. Respected dignitaries, distinguished guests, ladies and gentlemen, esteemed colleagues, friends, all protocol observed. My name is Bashir Muambi, and I do stand here to tell you how about our journey at the sites of training in Indonesia and how we felt. And I am honored to be the representative of the group. My speech is going to talk about from doubt to determination, a new era of vaccine development. What have we learned, and what did we know and what we didn't know? So in the first place, we had the doubts that vaccine manufacturing is not for the low and middle income countries, though at the end we did appreciated that it is possible with the determination, with the tools, the simplest tools available, anything is possible. In all the journey, we have been guided from when we landed in Jakarta up to Bandung. We went through quite amazing structures, amazing companies, firms that do produce vaccines. And really it was amazing to learn that such can be done at home. And while we thought, particularly when I was coming, that probably at this site we have only downstreaming facilities, I realized that we are also having facilities that can initiate vaccine development and way through to manufacturing, which is a plus. And once we keep this collaboration, it will be a very good and enticing idea for low and middle income countries, particularly the OIC member countries. In particular, vaccine production here emphasizes halal products. And yes, because if we need to support our communities, accept vaccines and uptake vaccines, then we have to emphasize inclusiveness to take care of the religions, cultures, and beliefs. I appreciate, and perhaps when I get back, I'll try to be an ambassador of promotion of vaccine production, because I'm well equipped with the knowledge to let the community understand that it is possible to have vaccines that adhere to halal standards. That is very good. And thank you so much for that. Lastly, I would want to thank OIC, Ministry of Health. Ministry of Health, you really were an amazing team. Thank you so much for all the organizations that care and everything. Then I would want to also thank COMSTECH for taking an initiative to have this blend and allow this happen. It's been a great journey. It's a milestone to learn from. The centers of learning, starting from Jakarta, Etana, Biopharma, UNPAD, BRIN, and others. It's been Kalbi. It's been really an amazing journey. We have learned a lot. In each and every facet we moved, we were saying, wow, this is now the end. I think this is the end of Indonesia. Then you move to another and you see other amazing different things. And it's a journey that has been so fruitful. We, therefore, my colleagues, have to remember the words of Dr. Yusuf during the training. Irrespective of the challenges, inadequacies in facilities, let us collaborate. Let us not end this here. We have to make this happen, and we shall be there. Please, when you go back, please do read. Please do collaborate. Don't sit back with this knowledge, because we have to support the communities. This will not only support public health facility endeavors, but also uplift the economies of our countries where we're coming from. Thank you so much, and may the Almighty Allah bless the hands of our deeds. May the Almighty Allah bless the path of where we are heading. Assalamu alaikum wa rahmatullahi wa barakatuh.
Once again, please give a round of applause for Mr. Vice. Thank you very much for the thoughtful feedback. And Excellencies, distinguished participants, now let us hear the remark from Excellency Mr. Aftab Saidi, the Senior Director HR and Administration of COMSTECH. Please welcome Mr. Aftab.
Bismillahirrahmanirrahim. Excellency, the Minister of Health, Indonesia, diplomats, representatives from OIC member states, DG Pharmaceutical and Medical Devices, Ministry of Health, DG Multilateral Cooperation, eminent experts, research fellows, valued guests, ladies and gentlemen, Assalamu alaikum. On behalf of COMSTECH, I welcome you all to the closing ceremony of third batch of INA-COMSTECH program for the research and advanced training in virology and vaccine technologies. It is indeed a pleasure for me to join you virtually for the closing ceremony of this very important program. It is wonderful to see this program up and running for the three consecutive years now. This year, we have 10 participants from 9 OIC member states who will benefit from this program. These participants are early career scientists, virologists, and public health researchers from Egypt, Indonesia, Malaysia, Pakistan, Tanzania, Somalia, Cameroon, Uganda, and Kazakhstan. In this closing ceremony, each of these candidates will also be represented by member faculty of their respective organizations, institutions. During this 1-month-long research fellowship program, participants learned many areas of vaccine technologies and its production, such as computational vaccinology, in vitro assay for vaccine development, nanolipid formulation for mRNA vaccine, antigen design for diagnostics and therapeutics, antibody, and last, micro encapsulation of natural products. The cycle of this program is a collaborative effort of COMSTECH, Ministry of Health Indonesia, Ministry of Foreign Affairs Indonesia, Biopharma, UNPAD, UNAR, last but not the least, Embassy of Indonesia in Pakistan. COMSTECH is extremely grateful to Ministry of Health Indonesia for its hospitality and its team for the wonderful work. The purpose of initiating this program is to enhance active participation of pharmaceutical industry, higher education, and research institutions of OIC member countries in the area of health research, particularly vaccine production. This training is aimed to enhance the research and development capacity of promising scientists, especially those at the beginning of their research career, helping them to foster linkages for further collaboration in the field of virology and vaccine technologies. This program provides OIC member states an opportunity to work collaboratively in the field of virology and vaccine technologies to reach their national goals collectively and individually. Vaccine production holds significant importance in the Muslim world for various reasons, including public health, economic development, and social well-being. Vaccine production in the Muslim world is crucial for addressing public health challenges, fostering economic development, and contributing to global health initiatives while aligning with Islamic values and principles. Building local vaccine production capabilities can enhance resilience, promote self-sufficiency, and ensure equitable access to essential healthcare interventions. We sincerely hope that this fellowship has contributed in the development and strengthening of the research and industrial capacities of the member states at the regional level through the contribution of eminent scholars from various universities and R&D institutions. COMSTECH hopes to continue its partnership with Ministry of Health Indonesia in the future with regard to this program and others as well. I hope participants will network through this training, creating ripple effect for science diplomacy in the OIC member states through COMSTECH. Thank you very much.
Rizka Andalusia, Director General for Pharmaceutical and Medical Devices. Excellency, Mr. Astaf Zaidi, Senior Director, HR and Administration, OIC COMSTECH. Excellency, Prof. Rizky Abdullah, Director of Research and Community Service. Excellency, Mr. Soleh Ayubi, Deputy Chief Executive Officer, Biopharma, other related ministry and institution, distinguished participants, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon. As we gather here today to mark the end of our one-month journey through the fascinating world of virology and vaccine technologies, I find myself both humble and invigorated. This fellowship has been an extraordinary experience, one that has broadened our horizons, deepened our knowledge, and ignited a collective passion for scientific advancement. First and foremost, let us extend our heartfelt gratitude to the incredible team of organizers, especially to the Organization of Islamic Cooperation Standing Committee on Scientific and Technical Cooperation, COMSTECH, the Ministry of Health of the Republic of Indonesia, University of Pajajaran, and Biopharma OIC Center of Excellence in Indonesia, who made this journey possible. Your dedication, vision, and tireless efforts have transformed what could have been an ordinary month into an unforgettable experience. From the meticulously curated lectures to the engaging hands-on workshops, every detail has been thoughtfully planned and executed. It is your hard work and commitment that have allowed us to immerse ourselves so fully in this vital field. Not to forget, a special dedication and thank you to Etana Biotechnologies, National Research and Innovation Agency, BRIN Laboratories, and also Kalbe Business Group for especially graciously accepting our visit. To our distinguished speakers and mentors, especially from University of Pajajaran, Institute of Technology Bandung, University of Erlangga, and University of Indonesia, we owe you so much. Thank you for generously sharing your knowledge and guiding us through complex topics with such clarity and enthusiasm. Your mentorship has not only enlightened us, but has also inspired us to push boundaries in our own careers. The insight you have shared will undoubtedly shape our future work and drive meaningfully progress in virology and vaccine technologies. A special thank you goes out to the support staff, students, and volunteers whose behind the scenes efforts ensure that everything runs smoothly. Your contribution, though often unseen, were crucial in creating an environment conducive to learning and collaboration. Over the past month, we have had the privilege of delving into the intricacies of viral mechanisms, vaccine development process, and cutting-edge research methodology. We have been challenged to think critically, collaborate effectively, and envision new possibilities for the future. The experience we have shared and the connection we have made will serve as a foundation upon which we can build our professional and personal growth. Moreover, our exposure to industrial setting was invaluable. The opportunity to witness firsthand the operations, challenges, and innovation within leading biotechnology firms and research institutions has given us a practical perspective on how theoretical knowledge is applied in real-world scenarios. Seeing the intricacies of vaccine production, quality control, and distribution has enhanced our understanding of the entire life cycle of a vaccine from concept to clinic. This industrial experience has provided us with a pragmatic viewpoint, emphasizing the importance of bridging the gap between research and application. It has shown us how our role as scientists can drive tangible impact on the public health and how collaboration with industry partners is essential for translating research breakthrough into viable solutions. One of the most profound takeaways from this fellowship is the importance of international collaboration. In our interconnected world, the challenges we face in virology and vaccine development are not confined by borders. The global nature of pandemic and health crisis necessitated a united effort where scientists, policymakers, and industry leaders from different countries come together to share knowledge, resources, and strategy, especially between the OIC countries. This fellowship has underscored the value of cross-border partnership and the exchange of ideas. It has demonstrated that by working together, we can overcome barriers, accelerate innovation, and address global health issues more effectively. This month was not solely defined by our scientific exploration. We also had the enriching experience of engaging in historical and cultural visits that were seamlessly woven into our program. So this education provided us with a broader perspective, connecting us with the rich history and vibrant culture of the region. Exploring this aspect has not only deepened our understanding of the local context, but has also highlighted the intersection of science with societal development. This unique integration of historical and cultural insight has enriched our fellowship experience, reminding us that the impact of scientific work extends beyond the laboratory and influences the world in profound ways. As we conclude this fellowship, let us remember that the end of this journey is not a conclusion, but a new beginning. We are now equipped with a wealth of knowledge and a network of like-minded professionals who share our commitment to advancing science and improving our global health. In closing, let me reiterate our profound appreciation for the organizers, mentors, and all those who have contributed to making this fellowship a resounding success. Your impact extends far beyond this wall and will resonate throughout our career as we continue to explore, innovate, and strive for excellence. Thank you all for being a part of this remarkable experience. Let us move forward with renewed enthusiasm and shared dedication to making a difference in the world of virology and vaccine technology. Before I end my words, allow me to read this pantun. Stars twinkle bright in the evening sky. Your kindness shine like the moon's soft glow. Thank you for all as time passes by. May joy and peace in your life always flow. Bright flowers bloom in the morning light. A shadow grow long with the setting sun. Thought we part ways, our heart remain tight until we meet again. My friend, be strong. Beras kencur, nasi kebuli, ikan gurame dan juga es. Selamat tinggal, semoga berjumpa lagi di majlis lain penuh kenangan manis. Ayam goreng, rendang nikmat, sate dan nasi, liwet yang lezat. Semua hidangan begitu hebat. Terima kasih kehadiran sahabat. Thank you.
Thank you, Mrs. Riza, for the sincere message. Thank you very much. You're pleased to return to your seat and for the great pantun as well. And distinguished speaker and participant, now let us hear remarks for this important occasion from Excellency Director General of Pharmaceutical and Medical Devices. Distinguished speaker, ladies and gentlemen, please welcome Mrs. Lucia Rizka Andalusia.
Distinguished expert, Mr. Aftab Zaidi, Senior Director HR and Administration COMSTECH, Professor Rizky and Professor Toto from University of Pajajaran, Bapak Saleh Ayubi, Deputy CEO PT Bio Farma, and also representative from Ministry of Foreign Affairs, Bulinda, representative from Indonesia FDA, representative from National Research and Innovation Agency, representative from pharmaceutical industry, and also alumnus from first and second batch of fellowship program, and participant of a third batch of fellowship program. Distinguished ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon from for of all us. First, I would like to congratulate all participants who have successfully completed this third batch of INA-COMSTECH Fellowship Program for Research and Advanced Training in Virology and Vaccine Technology. I believe many valuable insights and experiences in this fellowship program can further benefit your respective country. On this occasion, I would like to express my appreciation to INA-COMSTECH for entrusting Indonesia in a fellowship program for 3 consecutive years. I would also convey my gratitude to Ministry of Foreign Affairs, National Agency of Research and Innovation, National Agency of Food and Drug Authority, Pajajaran University, Erlangga University, Bandung Institute of Technology, University of Indonesia, PT Bio Farma, PT Etana, and PT KLB Pharma for their invaluable contribution to this training. Together, we can show a form of unique collaboration between ministries, agencies, and institutions that aims to strengthen cooperation between OIC member states in the field of vaccine research. Ladies and gentlemen, OIC members still face challenges in getting access to vaccine. Only around 70% of member states are vaccine manufacturers, and just 3.5% have products that meet WHO pre-qualification standard. This highlights that the vaccine research and manufacturing capacity in OIC countries is relatively low and unevenly distributed. Indonesia recognized that the important role of research in building of national vaccine capacity and also within the OIC countries' vaccine research and manufacturing ecosystem. As the health minister said at the OIC opening ceremony, if we have several centers of excellence to do research and development and manufacturers of vaccine, this will make a safer world for us and equitable access across the world in dealing with the next pandemic. Aligned with that, in this fellowship program, all participants have the opportunity to make a project on vaccine development. This project is expected to be a forum to discuss and build collaborative networks related to research of vaccine and biotechnology products. From this project, we hope all participants can apply this knowledge to enhance research capacity in their country. Indonesia is a nation that is currently enhancing vaccine research and manufacturing ecosystem, which has successfully produced 8 out of 14 antigen for national immunization program, and 4 of them has been WHO pre-qualification. And also we have mastering 3 vaccine technology platform, virus vaccine, protein-based vaccine, and also mRNA-based vaccine. Indonesia was also chosen as one of the recipient WHO mRNA technology and part in global vaccine clinical trial. We believe that progress and sustainable action can be achieved through partnership that pool resources, share expertise, and facilitate technology transfer. This approach is essential for enhancing vaccine capacity across OIC countries and ensuring equitable access to vaccine. This aligns with the goal of the OIC Center of Excellence on Vaccine and Biotechnology Product, which is to promote self-reliance in the supply and production of vaccine within the region. Ladies and gentlemen, after this fellowship program, all participants will return to their countries to implement the knowledge and experience, as well as build network among researchers from OIC member states. I do hope that you will continue to contribute to enhancing R&D and manufacturing activities of vaccine in your country and region to protect our people from disease and to promote well-being and pandemic preparedness. We also hope that OIC member and alumni from the first and second batches can sustain this program, strengthen research and manufacturing vaccine in OIC member states, and establish long-term global and regional research and development network, emphasizing the importance of collaboration with all OIC member states globally. I wish you have a safe journey back home, and thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Mrs. Lucia Rizky and Alicia, for your profound speech. Please kindly stay on stage as we will continue for the awarding session. And distinguished participants, we would like to invite all the participants, the program participants, to come on stage for receiving their certificate. I would like to invite Mr. Ebedigai Rowan from Cameroon, Mr. Sharif Magi Al-Maksud from Egypt, Mrs. Anissa Shetzer from Indonesia, Mrs. Jennifer from Indonesia, Mrs. Sandaga Shandi Bayeva from Kazakhstan, Mrs. Azima Abdul Wahab from Malaysia, Mrs. Nora Fiza binti Mat Isa from Malaysia, Mrs. Naila Shahid from Pakistan, Mrs. Zaira Rahman from Pakistan, Mr. Muhammad Abdirahman Omar from Somalia, Mr. Abdullah Musa Kitara from Tanzania, and Mr. Mwambi Bashir from Uganda. And we would like to also invite Professor Rizky from Pajajaran University also to come on stage with Ibu Rizka to give the certificate to the fellowship program participants. Distinguished ladies and gentlemen, while we're witnessing the awarding session, we also can watch the documentation video on the screen. And ladies and gentlemen, now we can watch the short videos of the activities during the third batch of INA-COMSTECH Fellowship Program for Research and Advancement in Virology and Vaccine Technologies. We can see all the activities that all the participants have been through during their time and their session in Indonesia. Congratulations to all the fellowship participants. We will continue for the photo session. I'd like to invite Ibu Rizka and also Prof. Rizky for the photo session that will be done on the floor. And for all the fellowship participants, please move forward. But still stay on stage. For the first photo session, we'll take a photo session with Ibu Rizka and Prof. Rizky. Maybe all the participants can come closer. And all the certificate, showing to the photographer. Let's do the countdown. 3, 2, 1. Once again, 3, 2, 1. Okay, let's give them our applause. And we'll do one more time for the photo session, but we will change the position for Indonesia, which is Mutran, and we will take a photo with all the participants. And for all the fellowship participants from the plus region, to the behind of Indonesia. We will change the angle, and all the participants in the room can please stand up or rise and come closer to the front. We'll take a picture together with all the delegations. Once again, please come closer and look to the camera. 3, 2, 1. Show a beautiful smile. 3, 2, 1. Okay, thank you. Please give a round of applause for everyone's here, and also for the fellowship delegation, ladies and gentlemen. Distinguished participants, please have a seat or return to your seat. Excellencies, distinguished participants, ladies and gentlemen, once again, International Harvard Webinar and Closing Ceremony, the top batch of Indonesia COMSTECH Fellowship Program for Research and Advanced Training in Virology and Vaccine Technologies, we have implemented it perfectly. We have reached the end of the session, but before we close all the agenda, we still have the Kahoot games that the committee already prepared 5 special prizes for those of you who will win the Kahoot game itself. The committee have prepared special Kahoot games for all the participants in the room and also to the Zoom meetings that can participate by using your desktop or also your mobile phone. Who knows that you will be the winner and bring home a special prize from the committee. The committee already prepared a special electronic money that will be transferred directly to your account. So please be ready before we end this session. Yeah, you can buy something for the dinner. Excellencies, distinguished participants, ladies and gentlemen, once again, International Harvard Webinar and Closing Ceremony for Batch of Indonesia COMSTECH Fellowship Program for Research and Advanced Training in Virology and Vaccine Technologies, we have implemented it perfectly. We believe this initiative will enhance knowledge and capacity of our researchers and achieve a betterment of our generations. And now we'll start the Kahoot games. And distinguished participants, I would like to therefore thank you very much for Indonesia and also our VIP for attending this special occasion. Yeah, please to continue your important and other agenda. Thank you very much. And now we'll continue with the special Kahoot games. Now be ready. Put the jumping 715773, and we will see who will be the winner and bring home a special prize from the committee. So now we have the current scoreboard Umebe, but it's still for the current, not yet. Only for the first questions. How many countries are members of OIC? Okay, what is the correct answer? We still have 11 seconds. Yeah. Okay, almost. Rama is the first one. Okay, it's a tough competition, yeah? They ready? Oh, I can see us audience over there. A lot of supporter. And the next questions? We still have other 6 questions until we can get the winner. Wow, it was something with 2,636. Be the first, I think. How many fellowship programs have been organized by Indonesia and country before this year? Wow, this is so easy. I hope everyone answered the correct answer. City still leading the scoreboard. Well, Umi is behind. Okay, how many countries participate in the third batch of Indonesia-COMSTECH fellowship program? Now it will be leading the board. We still have 2 questions left. Okay, this is the final questions. It's a tough question, I think. we go. And then the second place winner and the first one winner. So you like distinguished Professor and the former Wirema and the fifth winner of the paper. So congratulations for all the very educated kids. You are pleased to contact the committee behind there to get all the process. Make sure they transfer directly to your account. And distinguished participants, ladies and gentlemen, that is all the end of the session. We believe that this initiative will enhance the knowledge and also capacity of our researchers and also achieve betterment for our generation. With the end of the ceremony, that also ends of our today's event. And I am Santi Padaini on behalf of the organizing committee, extending our appreciation to each and every one of you for attending this closing ceremony. Further, and also please accept our sincere apology if there's inconvenience happen or any mistakes. We hope you have a great day and a productive day. Once again, thank you very much and see you again in another occasion. Greetings Halte Indonesia. To come on stage, and also on the organizing committee will have a photo session together. We will take a picture together, so to all the fellowship participants and also organizing committee kindly come to our stage. To all the policy participants, please kindly come to the stage, and also all everyone in the meeting venue, all the committee, organizing committee, we will take a picture together. Ayo kakak-kakak semua, ayo. Boleh yang masih ada di belakang, untuk semua organisasi komite dan juga partisipan, kita bisa foto bersama. Masih ada yang ketinggalan? Nah. Baik, semuanya siap dalam posisi. Kita dari apa dulu, Ibu? Gaya formal dulu, baik.